So guys, we shall be discussing regarding the Koch postulates, right? So Robert Koch, we have discussed about him. Now we shall discuss his postulates. What all postulates he have stated? Koch postulates are pretty important. Now what are these Koch postulates are? Basically there are five Koch postulates. So what are these Koch postulates are? The first important thing is that he stated that a specific organism is always found to be in association with the disease. Now, okay, first let me write and then I'll explain you. He stated that a specific organism can always be found in constant association with the disease. Now, what does he mean by this? What do you mean by this is that basically, for example, a disease is caused by a specific bacteria, right? So, there have to be an organism to cause a disease. So, he told that, for example, Mycobacterium leprae, okay, I am just giving you an example. Mycobacterium leprae will cause leprosy. It will not cause typhoid, right? So, Mycobacterium leprae causes only leprosy. It will not cause typhoid. In the same way, Vibrio cholera will cause cholera. It will not cause gonorrhea, right? So, gonorrhea is caused by different bacteria. So, he stated that for a specific disease, there will be a specific organism. Okay? Like for, let us say, leprosy, a specific disease called leprosy, for that we have got a specific organism called Mycobacterium leprae. Okay? So, Mycobacterium leprae causes leprosy. It does not cause gonorrhea or it does not cause cholera or typhoid and all. Right? So, every disease has got its own specific organism. So, that's what he told. Okay? Next important thing is that, okay, let me even write the example, guys, so that when you revise it later on, it would be easy. For example, M. leprae, right? Mycobacterium leprae causes leprosy, not TB or syphilis. Right? Second important example is Vibrio cholera. Causes cholera and not gonorrhea. Clear? Second important postulate what he told is that, second important postulate is, for example, let us say I have got some bacterial infection. Okay? Now, he told that, for example, if you got a bacterial infection, then if I take a blood sample, within that blood sample I will have that bacteria. Okay? Now, I can isolate bacteria from that blood sample which is taken. That is the second postulate he told. Okay? What he told is that, for example, let us say there is an individual over here, right? Now, in this individual, there is some kind of bacterial infection. So, I am taking a blood sample. From that blood sample, I can isolate that organism, whatever it is present in his body. Okay? So, I can isolate, I can extract that bacteria which is present in his blood. And after extracting that bacteria, I can inject it into a mice. When I inject it into the mice, it has to cause the same disease in the mice also. For example, let us say I am having mycobacterium, let us say I am having leprosy. Okay, so you have taken my blood sample. Now within my blood sample, you have very nicely isolated that Mycobacterium leprae. Once after isolation, you have injected that Mycobacterium leprae into the mice. So even in the mice also it should cause, the lab animal is the mice, right? So even in the mice also it has to cause the same disease like leprosy. Okay. Third important thing he told is that Once the mice gets this leprosy or whatever it is, when you take the sample from the mice, again you must be able to extract that organisms from the mice blood sample. Okay, so this is what he told. It is pretty simple, guys. What he told is that you can isolate, he gave another postulate that you can isolate the organism. You can isolate the organism in pure culture medium, from the blood sample. Okay, so the second postulate which he told is that you can isolate it. Okay, fine. After isolating, what will I do? After isolating, what can I do is that the third important thing is he told is that you can inject that isolated organism, whatever organism you have isolated from the blood sample, you can inject it into the mice, and you will see that even in the mice also you will find the same disease. Okay, so you can inoculate it into the lab animal. Lab animal is your mice, and that should produce same disease. Okay, now once after mice gets that same disease, again take a blood sample from the mice. Now when you take the blood sample from the mice, again you should be able to extract that same organism, because the same organism is causing the same disease in both humans and mice, right? So you can even extract that. So he told that you can re-isolate. First of all, you have isolated the organism from the human's blood, then injected it into the mice. Again you have taken the sample. Now again you are taking up that microorganism. You call it as re-isolation, right? So you can re-isolate the same organism. You can re-isolate the same organism from mice. Into what? Into a pure culture. And the last postulate which he told is that whenever any kind of bacteria enters into your body, bacteria has got antigens, right? Bacterial antigens. So when any kind of bacteria, if it enters into your body, it has got antigens. So bacterial antigens are in your body right now. So your body's immune system should produce antibodies against that. Okay, and obviously definitely your immune system will release antibodies against that antigen. Okay, so this is called as immune response. Okay, so he told that antigen must be able to produce antibodies in your body, must be able to produce antibodies in the body, and this is called as immune response. So overall, in the course postulates, what all we saw, guys, we have got something called as constant association. We have heard the term as constant association with the disease, right? We have also learned about isolation, right? And after that we have inoculated the organism, isolation, inoculation, and after that what we have learned? Re-isolation, right? So re-isolation, and finally immune response. These are the important points to remember. Immune response, right? So what all things we have learned? We learned that there is always a constant association with the disease, and after that you have isolated the organism, and after that you have inoculated the organism, and again you have re-isolated the organism, and finally antigen, whichever it is entering into your body, must produce antibodies, which is basically this phenomenon is called as a what? This is called as immune response. So these are some of the important coach postulates, guys. But so if an organism has to obey Koch postulates, as you know that organism has to cause the disease, right? And you must be able to extract that organism and again inoculate it and again re-extract it, right? And that organism must cause some kind of immune response in your body. Only then you can tell that this particular organism is obeying the Koch postulates. But there are always exceptions. So there are some organisms which do not obey Koch postulates. Organisms which do not obey Koch postulates. Now, what are those organisms which do not obey the Koch postulates? The first important thing, is that Treponema pallidum. Treponema pallidum. And what is the second important organism? Mycobacterium leprae. And the third important organism is Rickettsia. Third important organism is Rickettsia. The fourth important organism is Chlamydia. So, if you look here, these are the four important organisms that do not follow Koch postulates. Why? Because you cannot culture these organisms. I mean, you cannot extract in the pure culture media. Okay. So, I told you that organisms have to be, you have to extract them, right? You have to isolate them. So, you cannot isolate them basically. Okay. You cannot isolate them or you cannot culture them. So, for that reason, they do not obey the Koch postulates, and this is a very important MCQ for you. Okay, right. So next important thing is that the next important scientist which we are going to study is Paul Ehrlich. Paul Ehrlich. Right now, Paul Ehrlich, who is this scientist? He is called as the father of chemotherapy. Okay. Second important thing is that he is the only scientist who first discovered the antibiotic for syphilis. So he is the scientist who discovered what? Antibiotic. For which organism? Syphilis. Disease is syphilis. Okay. And he named it as salvacin. He named that as salvacin. Okay. And he is the one to describe about acid fast staining. You know, there are two types of organism, acid fast and non-acid fast, right? So you will learn it in the staining methods anyways. So acid fast staining. Fourth important thing is that he also told about the standardization method of a toxin and an antitoxin. Okay, so he also gave standardization of what? Toxin and antitoxin. Toxin and antitoxin. So these are some of the important things you need to know about the Paul Ehrlich. So guys, we have discussed Paul Ehrlich is the father of chemotherapy. So this can be a very important question. And the next important thing can be antibiotic, that is called as salvacin. So keep these things in mind. And we have also already discussed regarding the Koch postulates, right? So hanging drop experiment, Robert Koch. Solid culture media, Robert Koch, right? I told you to remember by the mnemonic SIDS, seeds. Okay. So this is a very important thing to understand. So these are some of the very important things regarding the introduction to the let us say the microbiology. So these are the scientists which you have to know, right? So they are important. And by the way, So guys, he is Anton van Leeuwenhoek. Anton van Leeuwenhoek, and after that, he is who? What is the name of this scientist? He is Robert Koch, who has given Koch postulates and all, right? And he is Louis Pasteur, and finally Paul Ehrlich. He is Paul. Clear? So he is the one who described about a disease called as Ehrlichia. Keep that thing in mind. Okay, so I think it is pretty easy. That is the reason why I haven't mentioned. So Ehrlich described about Ehrlichia. Clear? So this is all you need to know regarding the scientists in microbiology and their discoveries. So thank you so much for watching my lecture. Goodbye.